Okay, so our job today is to move into the world of organic chemistry. On Tuesday, we talked about general chemistry and talked about the fact that elements usually are identified by the relationships that they have with other elements. When I say valence, you should know what that means. So if I say atomic number, atomic mass, you should know what that means. Isotopes, and then we ended with the world of bonds. So that was last lecture, kind of in a nutshell. And so when we think of bonds, we're going to be talking about today how bonds are broken, how bonds are created, and how we build things. One of the big topics that we mentioned on Tuesday was that we have an example of a molecule or an element called carbon. It has 4 valence electrons and requires 4 additional structures for it to be stable. And when I say organic chemistry, it means that we have a carbon backbone. So when I took organic chemistry back in the day, I actually thought that this was a little better, a little more understandable, because it dealt with big molecules made of carbon. And some of these I think you have heard of, like lipids, which are fats, carbs, right? And then proteins, and then nucleic acids. So today I'm going to start off, before I even get into the lecture, by saying one of the ways that I would attack this lecture, parts of this lecture, is to create a table. Stay with me. So a table. I'm going to go ahead and say that the table that you create today, and I'll help you create it, will also definitely be on the exam. How's that? Now, I've had some students in the past who say, okay, I'm going to study that table, I'm going to memorize that table, and then they don't study anything else, and that's a disaster. Or I've had students who will study things but not be able to fill in the table. And that's like, I get my red marker, and I just put a big old question mark, like, what in the world? I told you it was on the test. So today, as we attack and we think about how to build large molecules and some of their properties, I think a table would be the best way to study. So we'll draw the table together, we'll fill in parts of the table, but I'll let you fill in the details and then study them on your own. Okay. We ended with a word, or some words dealing with water on Tuesday. We talked about the words hydrophilic and hydrophobic. Fearing water is phobia. Loving water is being philic. And we also mentioned a term, I believe, that when something has both, we call it amphiphilic. If that's a new term, write that down. Next class, we get to talk about cells, and we get to talk about components of cells. And one of the big characteristics of the outside of a cell, the membrane of the cell, is that it is amphiphilic. So hopefully that brings back some memories on Tuesday when I say amphiphilic. Something else that I would write down next to hydrophilic and hydrophobic, I would write the words polar and non-polar. So a non-polar substance is hydrophobic. Non-polar, fats, fatty acids. And hydrophilic water-loving are polar. So think about water. We said water is a great example of something that is polar, polar opposite. So polar and polar get together okay. Polar and non-polar, they don't get together very well. So oils and fats don't get together very well. We said that water was the universal solvent. Things dissolve easily unless they're hydrophobic into water. So we talked about electrolytes, and we said that in most cases, electrolytes are something that disassociate or are able to dissolve into water, and the importance of putting together an anion and a cation to form electrolytes. We talked about Powerade and how the label says ion 4, you know. So we know water is something that has to be considered. In the second semester, 152, there is an entire chapter, part of an entire lecture, that deals with water. Doesn't sound very exciting, but the fact that our cells are made up of water, most of the outside of our cells contain water, our kidneys are always worried about retaining water, our colon reabsorb. I mean, water is an important substance. Okay. Someone's pretty happy up there. So here we see kind of what I talked about, this relationship where we have fats and oils mixing with water, and you know, they just don't mix. Or in some cases, proteins, depending upon the size of a protein. Let's move into another big topic before we get into organic chemistry, and that is the topic of what does extra hydrogen protons or hydrogen electrons do, and how do they have an effect on biological solutions. So you may have heard of pH before. Sometimes just think of it as the power of hydrogen, pH. So hydrogen ions, here we go. When hydrogen ions are found to be in excess, then we say that we have either a proton donor or a proton acceptor. So an acid is something that releases hydrogen ions, and a base is something that accepts them. So when we think of acids and bases, most cases we think of the pH scale, and this is really, really important for anatomy, because as we talked about before, we have set points in a variety of places that deal with pH. So I think 7 is a good place to start. I would say that 7 is neutral, and below 7 is acidic, and above 7 is alkaline, or sometimes referred to as being basic. Sometimes I have students say, Mike, would it be worse to, dump a bunch of battery acid on myself, or spray myself with something like oven cleaner? And either one is horrible. So any extreme is dangerous. Any extreme is usually harmful to our cells. Something that I think is NCLEX worthy. Now, I apologize if you're not a nursing major, but if you're a nursing major, there's some things that you have to remember, and I mean just as facts. One of the things that I would like for you guys to remember is that CO2, carbon dioxide, is extremely acidic. And that's why we have to be careful with the amounts of CO2 that are found in our body. So if we have a lot of CO2 building up in our blood, in our body, then our brain says, guess what? Start breathing a little faster. Sometimes when we are completely unaware, completely maybe just sedentary, you find yourselves breathing a little faster. It's your brain saying acidity is building up in your blood. Breathe off that carbon dioxide. Or sometimes you might slow down your breathing because it might be getting too alkaline. So we want some CO2 to kind of get us back to pH of 7. So when we think of the pH scale, there's a lot going on, and most of it is happening behind the scenes, being taken care of, where we have no idea that it's being monitored and regulated. So that's pretty sweet. Now here's something that maybe you didn't know, is that when we look at the pH scale, it is something that we call a logarithmic scale. A logarithmic. Dominic, you're late no matter what. Wherever the class is. Look at it. Look at it.
Do you too?
Look at it. So let's talk about a logarithmic scale. What does that mean? That means if you've ever used logarithmic graph paper, then maybe the axes go from, look, 1 to 10 to, look, 100, 1,000 to 1, oh my gosh. Instead of like 1 to 2 to 3 to 4 to 5. So logarithmic means that we're making big jumps. So look, for example, when we say that we have a change in pH of just 1 on the pH scale, a lot of people look at me and go, what, so what? A drop of between 7 to 6 or 7 to 8? But if you drop a whole number or rise, listen, a whole number, that's 10 times more acidic or 10 times more alkaline. And if you drop, look, from 5 to 3, that's 100 times more acidic. Now that's impressive, or that's scary. At Xavier, I do a lab in the spring. It's my favorite lab. I'm sure it's breaking a lot of rules. You know, it's a urinalysis lab, and we use the students' urine. At Cincinnati State, we use fake. But since I'm in charge of labs, you know, I say I'm living on the edge. So kids pee in a cup like 2 times. And the first time they pee, we measure their pH. And if it's really alkaline, then I have those kids drink some lemon juice. Oh, they hate it. But it lowers their pH because of the acidity. And they'll drink some lemon juice to the point where they're almost sick. Then they'll pee in a cup, and it'll drop from like pH of 8 to pH 7.5. And they'll look at me like, Mike, you made me drink all of this and it only dropped half of a number. And I'm reminding them that, whoa, it's logarithmic. So that's a significant amount. That's why, hopefully you appreciate this. Look, when we look at blood, we're saying that the range of pH of blood is this. Now we know why it's that specific. Because if we have any changes in pH, it's a dramatic change or shift in the cell. Okay, so that's good. Let's talk about something maybe that you guys have experienced. When we think of maybe tomatoes, oh, that's a big one, or pickles, or wine, these are acidic. Some of you know that if you have maybe stomach problems, heartburn, that if you eat like red sauce, that doesn't work. Or if you think about this, eggs, that's pretty common. That is alkaline. But again, if I had to ask you which one's worse, stomach acid or oven cleaner, they're equally as bad. So don't just think acid is bad. Now sometimes acid builds up in places like our stomach, and some of us have issues just on the weekend. I mean, this weekend's a big weekend in Cincinnati, right? Taste of Cincinnati. Some of you are going to say, I am going to go down there and live it up. But before I do, I'm going to pop some Prilosec, or I'm bringing a whole handful of Tums, right? I'm going to prepare for the worst. But some of us have to take a Prevacid or something, a Meprazole or something, every day because we have problems with the cells that release acid. So much so that we have too much acid. And guess what Prevacid and a Meprazole are called? They're not called stomach solution pills. They're called proton, that's what a hydrogen electron is called, a proton pump inhibitor. I want you to write these letters down. You'll hear about this next semester, but some of you have already heard about it. This stands for gastroesophageal reflux disease. In other words, this is when stomach acid squirts up into your esophagus. Your esophagus is not designed for acid. So look where I'm pointing right here. This is where my esophagus is. If I have acid here, my chest burns. It's like there's acid. Oh, wait a minute. My heart burns. So I'm 52. I've never had heartburn in my life. I don't even know what that means. But my wife does sometimes. So if you have heartburn or too much stomach acid, you're told to take some proton pump inhibitors. That's pretty cool that you learned about that, right? Some of you will pop one of these this weekend, and you can use it as an anatomy review experience. Hey, let me tell you about that. Hey, let me tell you. Okay, so now let's move from some of the basics of hydrophilic, amphiphilic, hydrophobic, pH, to the process of creating and building. That's the definition of organic chemistry. So when we talk about creating or breaking, we have to look at something that either creates an ionic or covalent bond or breaks them. And in most cases, it involves water, that substance that we just talked about. So in some textbooks, you see something that looks like this, where A and B are the ingredients. Now let's call them the reactants. And C is the brand new result or product. So today, we're going to talk about how to assemble C. We're also going to talk about how to create A and B. So we can go both ways. Also, I want you to put a big star right here, because I'm going to guess at about 6:35, we're going to talk for about 10 minutes about this star and talk about how enzymes, here we go, can increase the production of C. So this reaction will probably take place, but it will take place a lot faster if we have enzymes. We'll talk all about enzymes, so don't worry. So now we know what reactants and products are. So if we're talking about building or we're talking about creating, there are three categories of reactions. There's ones that decompose or break down. You can write in your notes, maybe in parentheses, digesting is a great example of decomposing something. Synthesis is building. And then we have reactions that can go both ways. We call them exchange reactions. Now, if you're on your A game, we talked about metabolism. You guys remember the three parts of metabolism? Does anybody remember? Bria, hit me. Faster, Bria. Catabolism and anabolism, and then the third one? Fill her in.
Excretion.
Yeah. So when we look at these reactions, we can also write in your notes, I hope you are, that decomposition would be like catabolizing something, breaking something down. And to build, that's the definition of anabolism. If you can't figure out or remember the difference between the two, just remember the word anabolic steroids. People who cheat in sports who want to get bigger, they're building more and more and more. That's what anabolism does. Okay, so let's take a look at some examples of these different types of reactions. You guys are doing great. So let's take a look at a decomposition reaction. Notice the equation. It says that we have something big, A and B together, and we want to catabolize, digest, or decompose them into their building blocks. So here's something that is interesting.
What's worse? Going to Kroger and filling up gas and saying, ooh, 3 for $1 if I use my Kroger Plus card, 3 Snickers for $1, I'm doing it. Or going to Wendy's and saying, plain baked potato, please, no butter. Guess what? They're the same. Because if you eat a baked potato, you're going to break down that big starch molecule into what? Sugar. That stinks, right? Because you thought baked potatoes were healthy. Or we could say, let's take the individual building blocks and let's make something big. Like, look at this. Let's take some amino acids, you've heard of those before, and let's build something big like a protein. So this is anabolism, synthesis. We won't talk too much about exchange reactions this semester, but we will next. In fact, the whole process of breathing involves an exchange reaction. But exchange just means that, look, you can mix and match the reactants to get different types of products. A lot of times it's based on the need of a cell. So, yeah, go ahead.
For exchange, that's cellulatory respiration, right?
Yeah, respiration. Yeah, so it's involved with breathing and obtaining oxygen and getting rid of CO2. Okay, and then we have reversible reactions, and it says up here that this is the most common equation. I mean, do you have to study that or write that down? Heck no. Do you even have to recognize it? No, don't study that. But what I want you to recognize are the arrows. The arrows means that they can go, this reaction can go in either direction. So an exchange means we mix up the reactants to get different products. An exchange reaction says, if I am acidic and I want to become basic, or if I'm basic and I want to become acidic, I can go backwards and forwards. So that's pretty cool. We'll talk more about this next semester. Okay, so don't worry about the law of mass. Let's get into the reaction rates. Most of these conditions or characteristics of reactions are things that I think we would guess. We might remember back in high school that if you heat something up, you make the reaction go faster. That's one obvious thing. Another one is increasing the concentration of the ingredients. If you put more reactants together, you might make more products. There's always a limit, but that's important. And then you can see catalysts. Catalysts are something that we talked about a few minutes ago as being the star on the arrow. It means that enzymes also have a huge influence on how fast the reaction can take place. So when we think enzymes, we're thinking speeding something up. Now, each one of these different conditions can be abused. We can overwhelm our enzymes with too much glucose. And our cells will say, I can't handle it. And if the cells are in our kidneys, guess what they do? They say, I'm just going to pee it out. And so people who have diabetes or high blood sugar glucose, sometimes it's just identified by looking at glucose in your urine. Sometimes people can even smell it. Temperature, we know that most cases that cells love to be at body temperature. 37 degrees Celsius, 98.6 Fahrenheit. And we know that if we go above or below that body temperature, that set point, that can also cause problems. And some enzymes, sometimes enzymes get out of control. So there always has to be limits. But we're not going to call them limits. We're going to call them feedback mechanisms. Remember that discussion on lecture 2 of last week? Positive, making more negative, bringing us back to our set point. So all of these have to be regulated. Okay, so metabolism. Bria did a good job. She got 2 out of 3. She talked about catabolism being a reaction that causes the release of energy. We said decomposition. Also notice this word and circle this word, exergonic. That's a term that's important. It says that this is a reaction that releases energy. So by breaking something down, we have the opportunity to create something. But anabolism is something that requires fuel in order to build something. So it's endergonic. Exergonic releases, endergonic requires. So that's something to think about. There's a reaction that takes place in a lot of our cells on a daily basis called an oxidation reduction reaction. You don't have to know the details. This is not chemistry, crazy chemistry. This is anatomy and physiology. But you can see that an oxidation reaction gives up or releases. So we call oxidation an exergonic. And it says that reduction gains or requires input. So that's an endergonic reaction. Now get out your phone because this table doesn't exist in your book, but I like this table. This table tells you of all the reactions that we talked about, it organizes them into endergonic and exergonic. So snap a picture of that. Give me a second.
Can you pull the screen down so there's no glare? There's big glare in the middle of our.
Hi, Sam.
Oh, yeah. You got it, baby.
How about if I stand like this? Is that a distraction?
Thank you. Sorry.
All right, so there we go. If I say exergonic, you got it. If I say endergonic, you got it. Good? Man, this is like my mom used to phone. If I would take that long, you know what my kids would say? Man, Dad, you're old. Okay, so good. Let's move on. Something a little bit more exciting. Hey, this begins the world of our table. So I'm going to create a table over here. Why don't you write this down, and then I'll try to help you fill it in. I'll help you fill it in. We're going to start out with macromolecule over here. Then we'll say characteristics. We'll say function. And then lastly, we'll say example, look, examples. Okay. So that's the beginning of your table. Every year when I teach 151, I'll have students like obsessed with this table. This is only going to be 20 or 15 points out of 100. So it will prevent you from getting an A if you don't know it, but it's not the whole test. Everybody hear that? So don't obsess with this table. It's only 1 page of the test. And the test would be 8 pages. So this is not going to be the thing that you cover. Okay, so here we go. When I say macromolecule, we have to use some lingo. So the lingo that I want you to think about is the suffix mer. And so a mer is like a building block. Or think of it as one Lego piece if you're into building Legos. If we have one mer, we call it a monomer. If we have a two-mer, we call it a dimer.
Oh, that's many.
And if we have tons of mers, we call it a polymer. So what I would like for you to do is to say a macromolecule is a polymer. And after examples, let's make one more column. Oh, you're welcome. You are welcome. So you need to tell me, in addition to all of that stuff, you need to tell me what each macromolecule is really built out of. What are the building blocks? What are the monomers? Okay, so that's the table. I'll give you some hints. We're going to start with carbs. Then we're going to talk about fats. And then proteins. And then finally, nucleic acids. Okay. So far, so good. Welcome to organic chemistry. See, now, this is less scary than talking about protons and neutrons, because a lot of you have heard of these things before. So, smile. This is not going to be bad. Let's start out with carbs. And when we think of carbs, carbs belong to the organic classification because carbs consist of carbons, and we know that carbon has 4 valence electrons like we drew over here. We know that any organic molecule consists of many carbons in a row. We call them gigantic chains of carbons, or we say that they have a carbon backbone. Last class we said the difference between organic things and inorganic was the presence or the lack of a carbon backbone. So yay, a carb, a lipid, a protein, a nucleotide, they all have carbon. Okay, so that's good. That's a fact that we need to know. In addition to the carbon backbone, we said that things need to stick to these carbon valence electrons to make them stable. Sometimes it's other carbons. And sometimes it's things like an O and an H, or lots of H's, or an N and an, I mean, there's just, don't write this down, there's just, oh my gosh. There's just tons of opportunities for things to stick. And we call these things that stick functional groups. Now, if this was anatomy and physiology for medical school, I would say you need to know the functional groups. But this is anatomy and physiology for nursing, so I don't think you need to know them. So let me show you some, but you don't need to know them. Okay, don't study them. Don't say, I can draw them. Don't memorize these. You don't need to know these. Just remember what a functional group is. It's what makes an organic molecule unique. Because every organic molecule has carbon, but it's the functional group that makes it different. Okay. So here we go. Monomers and polymers. We said that a macro big molecule is nothing but a polymer. And polymers are many, many subunits stuck together. Or we can say many mers, many monomers. So we're going to call the monomer the building block or the subunit that creates these many mers. And each macromolecule has a different building block. So monomers are different. How do we create and stick together monomers to make a polymer? Well, it's called polymerization. And of course, guess what? It involves water. I want you to write this or circle this down, that when we remove water, we build. Look at this, dehydration synthesis. We build by removing water. And we break down or catabolize by adding water. So removing builds, adding breaks down. So look at the words. Dehydration synthesis builds, and hydrolysis is the process of breaking something down. Look at monomer number 1 over here. It has an OH. This is called a hydroxyl. Monomer 2, building block number 2 is a hydroxyl. If we remove, look, 2 H's, and an O, if we remove water, then all of a sudden they stick together and we create 2 monomers, a dimer. And if this continues, we create a polymer. This is polymerization by removing water. Okay? Removing water, dehydration synthesis. If we want to go in the opposite way, look over here. We have a dimer held together by an oxygen. We add water and we break them up and we end up with water over here. So now you know, this is very important, don't forget this, that water is involved with building and breaking things down. That might be something you learned today for the first time. Okay, so let's get into carbs. The first thing that I want you to write down in terms of characteristic of carbs is that they are hydrophilic. Carbs love water. Carbs love water. Carbs have a general formula, and I'm not going to ask you this, but they usually have twice as many hydrogens as carbon and oxygen. So for example, glucose, the one that we'll talk about the most, has twice as many hydrogen and 6 oxygen. Most carbs have a root word called saccharine. So the root saccharine can be modified. If it's one saccharine, we call it a monosaccharide. If it's a dimer, we call it a disaccharide. If it's a big one, we call it a polysaccharide. Okay, so now it's starting to kind of make sense. How do we create polysaccharides? It's all about water, right? Now, you do not have to study any of these pictures. You don't have to give me even any examples. So just breathe here. But I do want you to know that simple sugars are the building blocks for polysaccharides. So if you want to go all the way over here, you can say that the monomer is a monosaccharide. And we talked about this being hydrophilic. Or we could say polar. Okay, I'm not going to fill in the entire table, but you're getting the idea. Maybe have that table sitting to the side. So here are some examples, some monos. Glucose, oh that's a big one. Galactose and fructose. And we know that when we eat complex carbs, that it is ultimately broken down into glucose. Carbs have gotten a lot of press lately. You know, you hear about these people who are carbophobic, or people who love things that are carbolicious, right? And I think, if I can say this, I think that all of us, I'm assuming, and I'm throwing every American in this room not in the same boat, eat way too many carbs. I mean, way too many carbs. Now, we need carbs. All of you hear that. We need carbs because our brain is a big consumer of carbs. Our muscles need carbs. Some of you might even remember the glory days of high school where you would go and say, we're going to carb load before the big game or the big cross-country, whatever. Every marathon in the United States has a pasta dinner the night before the marathon. So there's something about carbs that provide energy. So when we think of function, they provide, and underline this, short-term energy. Short-term. So if I eat 2 out of the 3 Snickers from Kroger and put that other one in the glove compartment and then say, oh, no, I can eat that. If I eat 3 Snickers on the way home, or if I'm better and I say I'm going to eat a baked potato from Wendy's, it doesn't matter because they're ultimately going to be broken down into glucose. Your liver, stay with me, your liver stores the glucose. And it says, I'm ready for you to do something. But if you don't do something and you keep eating more, some of us will say, I would like a baked potato, and I would like this, and then what's for dessert? You know? Your poor liver says what? That's enough. Time out. I can't handle all of this glucose, so I'm going to store, ready, glucose in a long-term storage called fat. So the reason why most Americans are overweight is because we have too many carbs. There is no reason, remember this for the rest of your life, there is no reason when you make the healthy choice of going to Subway that any of us need to eat both pieces of bread. That is just like a sin. There's no way. All of those carbs, unless you're saying I'm going to go run a 4-hour marathon, and no one's doing that, all of that bread gets broken down into glucose. So that there is some legitimacy to saying that you should watch your carbs. I'm not a crazy psycho person that says no carbs, you know, keto diet, but I'm saying we all should watch our carbs. Because unless we're doing something, they're going to get stored as long-term storage as fat. All right, let me get off my high horse. When we look at disaccharides, these are 2 monomers put together to make a dimer. You don't have to remember any of these. But you can see that table sugar is a combination of glucose plus fructose. Or look at this. This carbohydrate in milk is what makes it sweet. A lot of people have a hard time breaking this down. We'll talk about lactose intolerance later. And then you can see maltose is usually associated from plant material. So again, plants, even though we all should eat them, fruits and vegetables, some plants have a lot of carbohydrates. Now the polysaccharides, I think, are the ones that fall into the category of gigantic carbohydrates. Polymers, or polysaccharides. One of the ones that we've talked about already is starch. It's a large polysaccharide. It's a member of a carbohydrate classification. Its job is to store, ready? Store glucose. So when we think of function, we're saying that it's a storage structure in plants. So we eat potatoes and whatever that have starch. In humans, the form is called glycogen. Glycogen is what we store our glucose in our liver. So glycogen is the human storage tank for carbs. Starch is the plant version. Now, one of the benefits of eating plant material is that around plants, I'm not going crazy with this, but around plants, we have structures called cell walls. And cell walls have this stuff, and we don't have the material or the enzymes or the microbes that can break this down. So this becomes fiber, or my grandparents would call it roughage. So don't nod and don't, in fact, I'm gonna close my eyes when I say this, because I don't want to see anyone saying yeah. But we all know that when we eat corn, okay, stop. Don't, no, I'm not looking. So why does corn come out looking like corn? Because it's surrounded by cellulose and our body can't break that down. That's a good thing. We'll talk about different types of fiber a little bit later. Yeah,
I
John.
have a question.
Hit me.
When it, like pertaining to vegetables and stuff, because I think there's like different vegetables other than corn that come out looking similar to how
Like
we eat.
peanuts and whatever, yeah.
So, but like, doesn't that mean we don't digest it well?
Well, corn is surrounded by a very thick coating of cellulose. So we get maybe the benefit of the starch inside of the corn, but we can't digest the outer material. So things like, maybe like strawberries or apples, you know, we don't really digest the skin very well of an apple, but the internal part we're able to digest. So it just means that the kernels of corn are just surrounded by cellulose. So it depends on how much cellulose is present. That's the answer.
Right.
Okay, let's talk about lipids. So let's look at our chart. First of all, with carbs, characteristics, hydrophilic. You can even say that in terms of characteristics, they have the same structural formula. Twice as many hydrogens as carbon or oxygen. Function, we said short-term storage. Examples, you could talk about monomers, dimers, and polymers. You could talk about glucose, fructose, or starch. I mean, you could fill that whole table up with a lot of examples. So make sure you put at least 2 on the test, okay? That's why I said S up there. First thing that you're gonna write in terms of characteristic of lipids is that lipids are nonpolar, hydrophobic, meaning they do not mix well with water. When we see some of the other characteristics, it says that they are less oxidized. Remember that reaction? Which means that they are harder to catabolize. We all know that. If we're trying to lose weight and we walk 30 minutes on a treadmill, it says that we didn't even burn enough calories that are in one Snickers bar. 30 minutes, not enough for one Snickers bar. It takes a lot of effort to break down fat. Fat is designed to be like what we need in times of starvation. It takes a serious effort to break down fats. It's less oxidized. Look at this. Here are some examples that you can put in your table. You don't have to put all of these, but here's some examples. We'll talk briefly about some. Circle fatty acids because those are the monomers. Ready? Fatty acids are the monomers of lipids, the building block. You probably heard of triglycerides if you're into your blood chemistry, had your blood taken. We're going to talk about phospholipids even more when we get into the cells. We talked about phospholipids being amphiphilic. Echinosoids, we'll briefly talk about. Same thing with steroids, that's more of a second semester thing. So take a look at this gigantic organic molecule, a fatty acid, the building block. Look at how many carbons, are you kidding me? Look at all of the hydrogens. I mean, this is what we call a saturated fatty acid, meaning that it is completely satisfied and there's very few, if any, double bonds, covalent bonds. So this is as stable as it gets, and in fact, when we talk about saturated fats, write this down in your notes, that saturated fats are solid at room temperature. Unsaturated fats are liquid at room temperature. So when we think at the difference between these different types of fats, we can say that unsaturated fats are probably easier to break down if they're in liquid form, which is true, but all fats are hard to break down. It requires many enzymes, even this stuff called bile, which some of you may have heard of before. My wife, when we first got married, was cooking with vegetable oil, which we thought was okay. Then she went to canola oil. And then for like the last 5 years, she cooks with nothing but olive oil, because it has the highest amounts of unsaturated fats, the ones that are better for you, the ones that you can break down. But every once in a while, we'll drive up to Cleveland, that's where my son went to college, and we'll go through Amish land, you know, and it's hard to drive through Amish land without saying, I'm gonna get a pastry, and they cook with like big sticks of butter, you know, lard, animal fat. Oh, that's saturated fat, that's the worst kind. Man, that tastes good sometimes. Okay, so function. We know the characteristics. Function, we're saying long-term energy storage. Fats are great for insulation and shock absorption. Some people think that fat should be eliminated. That would be incorrect. So there's always fat necessary, especially around vital organs. If you looked at my heart and you looked at a Kenyan marathon runner who was a vegetarian, there, throw that in there too, and you dissected our hearts, both of our hearts would have fat around it because it's a great shock absorber. Do you know, or did you know, listen, that there is nothing supporting our kidneys? Nothing. I mean, if I would say everybody stand up and run in place, do jumping jacks, what keeps your kidneys from like bouncing all over the place? They are impacted, surrounded by fat, tons of fat. And that's not saying you're obese, it's saying that that's biologically important. Okay, so good. Let's move on to some examples, and we'll talk more about this on Tuesday of next week, so you don't have to go crazy, but when we look at the components of the plasma membrane, the outside of the cell, we have regions that are hydrophilic, areas that are hydrophobic, and we say that they are amphiphilic because they contain both. Good, we'll hit this again hard, so don't worry about it. Don't write anything down here. In fact, if you have your outline, you can just put an X through this part. Echinosoids and steroids and hormones, these are things that we're going to focus more on next semester. But most people associate cholesterol with something being bad. But here's something that's worthy of writing down, is the fact cholesterol is the mother, listen, the mother of all steroids. So when you think of how testosterone is made, how estrogen and progesterone are made, it's all derived from cholesterol. So that's important. On Tuesday, when we talk about cells, we're going to say that cholesterol is also important to be inside the membrane. So it would be incorrect on the exam to say a completely healthy person would have 0 cholesterol. That would be bad. But if cholesterol is found in weird places like the arteries and places like that, then that's when it gets worrisome. But we need cholesterol. Don't forget that. Third, and our next component is the one that we're going to talk 10 minutes about. So we only have 15 minutes left. 10 minutes are going to be devoted to proteins. And I would say that you will need 5 pages to talk about functions for proteins. Now, you don't really need 5 pages. Just write down a few. But proteins, as it says up here, are the most versatile. And it says that the building blocks of proteins are amino acids. So now you have monosaccharides. Look, you have fatty acids for lipids, and you have amino acids for proteins. Okay, so I'm going to draw some stuff. Let's see. Man, I don't remember your name. What's your name? Yeah, what's your name?
Me?
Yeah.
Okay.
Oh, yeah. Can you tell me, Mike? So when we think of proteins, I want you to write these numbers down. Dude, on. So this is the different The basic building block level is called the primary level of organization, which means we're gonna take the building blocks, the monomers, and we are going to polymerize them, assemble them into long chains, and they are held together by very unique bonds, ones that we've never talked about before, called peptide bonds. Underline that. Peptide bonds are only found holding together amino acids. So when we talk about the beauty of proteins, some of us say collagen, maybe. Well, that's in our face, that's in our skin, that's in our joints. Some of you who've taken biology might remember myosin and actin for muscles. But when we think of the most biologically significant aspect, in my opinion, ready? Proteins are enzymes. Oh, I told you we'd come back to that. So what I'm drawing right now is how to build an enzyme. Something impressive, a polymer. So the first step is to assemble amino acids by peptide bonds. The second step is to have them fold like this, you know, into crazy shapes. In fact, it's called folding. How do we get the folding? We use things like van der Waals forces. Remember those very temporary attractions we talked about at the end of class on Tuesday? And what happens is that it creates something. It goes from this folding into this gigantic, look, glob of gum or a popcorn kernel. And we call this the tertiary structure of a protein. So you can draw a big popcorn kernel. And let's put 20 stars by the tertiary form of proteins. Because it's this structure that's biologically important. Because when we have a glob, stay with me, we have one, maybe more of these gigantic spaces. And it's these spaces that interact with, ready? Reactants. It's all coming together. Let's give the name to this space. It's called an active site. Don't forget that. That's worthy of circling and starring. Star, circle, underline. So the active site is the space in the protein. Think of it kind of like a lock. And the reactant, well, if a reactant, let's see if I can do it. Rear. So if a reactant has this part that I'm shading, look, that looks kind of like what would fit into here, then this is the key. You could say the reactant, but we're going to use some lingo here that's really important. We call it the substrate. So, let's rewind. Proteins have thousands of functions. When we talk about proteins acting as an enzyme, we talk about the three-dimensional glob. The three-dimensional glob creates spaces called active sites that are very specific to substrates. Not all substrates, usually just one. If I could fast forward till about, I'm going to guess, July 20th in this class, we're going to talk about medications for mental illnesses. That's a tough one. Or we'll talk about ADHD or something like that. And when we talk about mental illnesses and things like that, a lot of medications try to mimic, look, the keys, the substrates that fit into these enzymes, to block enzymes from working. Because some mental illnesses is an overproduction of something. Some of you know what I'm talking about, because you know what the drug does that you're taking. So there you have it. That is a three-dimensional glob that's biologically significant. You may ask, what about the quaternary structure? This is a structure that is not biologically. In fact, it's saying that it went too far. So in most cases, we're going to stop right here. We're going to say that tertiary is the most important. Okay, so that's important. Let's talk a little bit about the stability of these enzymes. We know that temperature, right? Concentration, all those things that we talked about. And Carter, can you turn those lights off again? So when we talk about tertiary, we can say that according to, thank you, one more on. You leave all the lights off, I'm gonna fall asleep. Okay. So here we have the amino acids. It's folding, and that's their that's not good. That's their version of a popcorn kernel. I don't like that. If we expose a protein to something that is harmful, like high temperature, difference in pH, we may cause it to go backwards in time and go from a tertiary look back to its primary form. All of you in this room know that if it goes from this big glob to this big strand of beads, there's no more active sites, which means the enzyme doesn't work. So use the lingo on the test. Underline this word. When enzymes fall apart, why? Because of pH or temperature, they denature. If I give you a situation like, oh man, this kid has a temperature of 108, what's happening to his enzymes? Don't write they die, okay? Say they denature. You do not have to write all of these down, and we will talk about all of these eventually. But in your function, look, in your function column for proteins, we can say they're involved with structure and communication, transport, enzymes, oh, we're not finished, recognition, movement, cell adhesion, oh, it can go on and on and on. So, for the exam, just know 2 or 3, put 2 or 3 in your table and call it a day. I promise that we will talk about every one of those at a later time. You will know, you will become experts on those. But for the first exam, just say, you're good with listing 2 or 3 of those. We said that enzymes work on substrates, and sometimes the substrate is what gives the name of the enzyme. For example, look at this. There is a carb called amylose. The enzyme that digests it is called amylase. So whenever you see the suffix ase, you get excited because you know it's an enzyme. Okay, so let's take a look at an example. Let's see. Maybe not. I thought I had a better picture. Let's take a look at this. This is their three-dimensional glob. Here's the active site. Here are the substrates that fit into it. And notice that once it kind of combines, it's able to spit out a new product. So let's go back about, an hour ago? Here is A and B. The enzyme is grabbing a hold of A and B. And here is C. I told you we'd come back to it. So enzymes can do this quick, and the cool thing about an enzyme is that you can recycle them. We can reuse them. And you can make a ton of C, make a ton of product. So the cool things about enzymes is they're reusable or recyclable. They do it really fast, but they're very fragile. Notice that temperature and pH could cause them to denature if they're not in the correct environment. Sometimes, and you can put an X through this in your notes because I'm not going to ask you about cofactors, but sometimes enzymes require a little help. called cofactors and coenzymes to bring the substrate and the enzyme together. Don't worry about that, talk about that later. So that's good, we're done with proteins. We spent 14 minutes talking about that, was good intent. So we'll spend 30 minutes talking about nucleic acids. And then we're gonna say we're done for the day, we did great. So our last category of a macromolecule are nucleic acids. And in big and bold are the building blocks called nucleotides. So let's fill in that last column again. Look, carbs, monosaccharides, lipids, fatty acids, proteins, amino acids. Nucleic acids are made up of nucleotides. And we're going to talk about the details of what's necessary to be a nucleotide. And we're going to talk about examples like, look at this, ATP, DNA, RNA. So when it comes to examples, you can write all of this down. But in terms of their characteristics, you can say they either carry energy, look at that. They are the structure that holds the genetic information, and they're involved with making proteins. So the characteristics are the fact that they have these three components, and the functions are all three of these: energy, genetics, and proteins. So when you look at this table now, you have a pretty good idea of what each macromolecule is, what is their monomer, their characteristics and function, and examples. So do me a favor and polish up this table, and then show it to me, if you would, next week during lab. So I'm not going to grade it, I'm not going to look at you if you don't have it, but you might just say during lab, Mike, here's my table, will you take a look at it so I know I'm studying the right thing? Will everybody do that for next Tuesday and Thursday? Okay. All right, so have a great 3-day weekend. If you have lab, start at 7 o'clock. And listen, let's say this, lab today could be a 45-minute lab. It could be